Okay, so good evening. My name is Ankita. I am your TA for this live interaction session. And as you know that in each session, we take up the topics that were discussed in that week's lectures, and we go through some in detail discussion about the same, and we look at some of the assignment questions for that week as well. So, as you know, the topic for this week was basically centered around the nervous system. So, what we'll do is we'll go through the lecture notes and go through each of the topics that were discussed. And if at any point there is some confusion, you can bring it up. And this is the third last session for this course. We will have 2 more weeks for week 11 and week 12 discussions. I will not be discussing any of those topics because those assignments are obviously still open, and these will be taken up in the upcoming weeks as well. So this week we'll restrict the discussions only to the nervous system. So for the recap, we basically had 3 lectures for this week's topics, and under that, as we begin every lecture, as well as has been discussed by Dr. Vishal Trivedi as well. We have started off with the classification, then we moved on to evolution, etc. And currently we are looking at each of the physiological processes that are operational in allowing an organism to be functional in order to help them to survive, right? So under that, we have taken up previously the circulatory system, the muscular system, and now we are coming to, and the digestive system as well. Now we are coming to the nervous system for this week. So as you understand that one of the major distinguishing features of humans as compared to that of other organisms is their ability to be to have cognition, because we are able to understand, we are able to think, and then we are able to make some decisions and execute an action based on the decision that has been made. So the reason we are able to do that is because our brain is coming in, our brain is capable of processing all of the information that comes in from the environment, from the from our own body, a decision. So the nervous system has an immense role to play in allowing an organism to be, in allowing humans, in our case, we are talking about humans for now, in allowing a human to be, to have the ability to think, to have the ability to respond, and obviously survive, right? So it is the, you can think of it as the coordinating center for all of the different physiological systems that are operational, which includes the systems that we have already discussed. So like it has been mentioned here, it coordinates physiological functions in humans. Um, Nervous tissue, it originates from the ectoderm. So why is this important? Is because if you have studied anything about the way development takes place, like after fertilization, from there you become a zygote, and then that zygote keeps on developing through the morula, gastrula, blastula, all of these stages. The first tissue that is observed while an organism is developing is the nervous tissue. So the notochord, or we have discussed notochord previously as well. It is one of the distinguishing features between vertebrates, invertebrates, chordates and non-chordates. So that is the first tissue which develops. So you can understand how relevant it is in allowing an organism's development to occur properly and to constitute its being, to make it really alive. It is a living being because this tissue has developed. And in fact, you can even compare it to end stage patients, you know, where if an individual who has suffered some sort of trauma is no longer able to respond to a stimulus, they are said to be brain dead, which means that they are in a vegetative state. So despite all of the other organ systems or organs being functional, like for example, their heart might be working, their liver, kidney, etc., all of that might be working, but their brain is no longer functional. So that is making that person basically, as crude as it might sound, it is that individual is often considered to be a vegetable in loosely used medical terms. So that is, I'm just stressing on this to really bring to your attention how relevant and how important the system is. So the origin of this tissue, as we have discussed, while a fetus is developing, is from the ectoderm. So if you remember, we had discussed during classification about how living organisms can be diploblastic, triploblastic, etc. So in triploblastic individuals or organisms, you have the endoderm, ectoderm, and the mesoderm in between the two. And from each of these three germ layers, what you get are each of the organs and organ systems, which ultimately constitute the entire living organism. And among these, nervous tissue originates from the outermost layer, which, as we already know, is the ectoderm. And this has the special feature of receiving stimulus, like I said, either from the environment or some stimulus from within the body. And in response to that stimulus, it is able to transmit that stimulus or that message, and the individual is able to execute a response. So the entire nervous system, we call it a system because it is actually quite, you can say, widespread. It's not localized to one particular region, and it has these three major branches. Of course, the central nervous system. Why central? Because it is you can think of it as the core, or as the basic um, unit, which is needed for an organism's nervous system to be functional, which is of course the brain and the spinal cord, like we discussed in depth about how important the brain is, right? And after that comes the peripheral nervous system, which, as you understand, when we say periphery, it basically means the surroundings, or the technically it's the boundary, but basically it's not the central part. It is an extended portion or the surrounding portion, and that includes the rest of the body, the nerves in the remaining body, which are which majorly include the cranial nerves and the spinal nerves. And finally, we come to the autonomic nervous system, which is, you can think of it as a reflex or involuntary system. Autonomic means something which is not really in control, which is a sort of default response, or an involuntary response, which is elicited by the system. And then under that, you have the sympathetic and the parasympathetic response, which can be executed by the ANS, or autonomic nervous system. So these are the major branches, like you're already familiar with it. So first in-depth discussion was carried out about the central nervous system, as you already know, and we have talked about it having these two major components, the brain and the spinal cord. So if you have gone through the lectures, you already know that basically, so, if you have read about the skeletal system, you would know that the skull, or the protecting cover of or the protective covering of the brain, basically the skull, is the strongest. It's considered to be the strongest bone or set of fused bones within any human body. It's called as a fused bone system because it's not one complete bone. There are several, like if you've ever seen a model of a skeleton, or if you just Google some images of it, you will see that it has multiple divisions, but they're all sort of fused, and they constitute a composite sort of system, the entire skull. So this set of fused bones has the immense responsibility of protecting the brain, which is why its strength is so immense. So you can survive if you break any other bone in your body. Most bones in your body, even if they're broken, they would get repaired and you would survive. But if the skull is damaged, severely damaged, then the brain, of course, gets it gets affected, and as a result, the individual will suffer from a lot of problems. So it protects the brain from external pressures, and it is basically it basically has a triple membrane or triple covering of connective tissue, which are called the meninges. So cerebral meninges are basically these layers of connective tissue. I have a better image for your understanding here. Yeah. So this is basically, you can think of it as a cross section of an individual's head. So what you see here, the outermost layer, is the scalp. It is like if you touch your head, underneath the hair, what you can see, what you can feel or touch is the scalp. Underneath it, you can find the skull, like very, you can see like little dots, which are indicative of the bone which is present, the connective tissue, the bone which is present, and that is the skull. Underneath it, you can see three layers of connective tissue within which is the brain. So this entire of yellow, of white kind of colored structure is the brain, the cerebrum part of the brain. Whereas these layers of connective tissue, they have that cushioning effect or that protective effect on the brain tissue, so that even if some sort of like superficial damage occurs to these skull somehow due to some accident, some trauma, even then the brain would not get very severely damaged, or you won't, some tissue or some nail or whatever external pressure might be coming in, it does not directly affect the brain tissue, because that, if you think about it, is irreparable, and it's not something the living organism would be able to afford. So after that, like we were discussing, the three layers of connective tissue, those are the dura mater, the pia mater, and like you saw here, the dura mater, the pia mater, and the arachnoid. These are the connective tissue which are present. Among these, the pia mater is the innermost layer. It is the thinnest. And when we say highly vascular, it means it has the maximum blood flow. Very obviously, if you think about it, one of the major reasons why people die of being in conditions where there is very low oxygen, or basically you can think of it as anoxia, or low oxygen or high CO2 areas, is because the brain does not get oxygen. And why do you need oxygen, if you ask? It's because metabolism, with the body metabolizes whatever nutrients it gets by the virtue of food intake. It requires oxygen for that, most of it, because aerobic oxidation is how we or aerobic metabolism is how we majorly survive, how we produce energy. And the brain requires the maximum amount of energy within the body. So oxygen supply, and continuous rich oxygen supply, is extremely crucial for the brain to function properly. If I give you a very simple example, like you see that often, uh, you know, people at older ages, when they have some sort of ionic imbalance, they, like, if you think about it, you might have a grandparent or somebody who might have low sodium, high potassium, these kind of difficulties, right? Under such circumstances, these imbalance usually leads to such individuals suffering from um, they start behaving differently. They do not respond in a normal manner. They become insensitive to what is being said to them. They lose track of what is happening around them. So that is not, I'm not talking about oxygen, I'm just talking about how these little nutrients, these micronutrients or gases, how they regulate how the individual will function. They stop responding properly, which means that that person is no longer who you used to know, because of some sort of nutrient deficiency or oxygen deficiency or something of that sort. So if you just sum it up, you can think of the brain as a very, very sensitive organ, which is very responsive to even slight changes in its environment. So the pia mater, like I said, is thin, delicate, it's highly vascular, and it is quite firmly adhered to the brain. The second, or the middle layer, is the arachnoid membrane, which is thin, it's folded, and it is sort of a sort of folded structure present in front of the cranial venous sinus. Why does it have these foldings or these infoldings? It helps to reabsorb cerebrospinal fluid, or CSF, in around the brain. Finally, the outermost layer, which we said previously, is the dura mater. It's very thick, it is strong, and it's non-elastic. And when we say that it is strong, it is because of what it is made of. It is made up of collagen, which is, is one of the strongest materials within the living body. One of the strongest. Enamel, teeth enamel are the strongest, but yeah, it's one of the strongest materials. And what is the remaining space that is present between the dura mater and the arachnoid? That is the subdural space, which is filled by the serous fluid. And a space between arachnoid, pia mater, it is the subarachnoid space, and it is filled by CSF. You can also get an idea about this from the figure as well. So like you can see, here is the subarachnoid space and here is the subdural space. So you will be getting access to these slides as well. So you can go through them for your better understanding as per your requirement. Coming back, we are looking at the brain and continuation. So we have mentioned the CSF or the cerebrospinal fluid, which is much like lymph. You have heard about lymph, right? When we talked about immune system and we understood how lymphatic fluid is responsible. You've heard about lymph nodes as well. You often get, you get your neck checked by doctors when you suffer from some sort of fever, etc. That is because there is a lot of inflammation in the lymph node. So lymphatic fluid is responsible for it. So visually much alike to the lymphatic fluid is the CSF. It is alkaline in nature, and it sort of is the if I put it a little vaguely, you can think of it as the shock absorber, which is present around the entire central nervous system, not just the brain, but also the spine, hence the name cerebrospinal fluid. It's around the cerebrum and it's also around the spine. It has immense role to play. Individuals often, some patients, some people who suffer from a little how should I put it? Like those who have some sort of reduced levels of CSF in their along their spine, along their vertebral column, they suffer immense pain because their nerves, they often tend to impinge one on top of the other, and that leads to a lot of back pain and many other extended diseases as well. Usually these are more of age onset disorders, but you can find them in young people as well nowadays. So what it exactly does, so these are extended functions, right? If things go wrong, then it leads to disorders. But then what is its function? Why is it present? So of course, it provides support. It protects the brain. It acts as a cushion, a shock absorber. But what it also does is it helps in the exchange of metabolic substances between the brain and the blood capillaries. And it is present in the brain ventricle, in the subarachnoid space, and like I said, around the spinal cord. So that is the connective fluid which is present there. Now, if we just come to the brain, so we discussed the outer layers, we talked about the fluid which surrounds it. Now we come to the brain. So the brain, like you already might have studied, it has three parts. Naturally, the forebrain, the midbrain, and the hindbrain. So these are the common terms, but for your better understanding, you must remember the more, you can think of them as the more traditional or more scientific terms. So the forebrain is the prosencephalon, the midbrain is the mesencephalon, and the hindbrain is the rhombencephalon. So each of these major parts, they also have subdivisions, right? So first of all, the forebrain, what is it? Forebrain, as you understand, is the front part of the brain, right? It has the cerebrum, it has the diencephalon and the olfactory lobes. The midbrain, the middle part or mesencephalon, it has the corpora quadrigemina and the crura cerebral. Thirdly, the hindbrain or the rhombencephalon at the back of the brain, which is quite close to your neck, to the back of your neck. It has cerebellum, pons, and medulla oblongata, the three most crucial, I wouldn't say most crucial, but they have immensely important roles to play in cognition, in normal functioning of a human being. It's also called as the brain stem or the hindbrain, and from there it connects to the spinal part of the central nervous system. So first, the prosencephalon or the forebrain, like we said, it is made up of these three parts. So the cerebrum, which is the major component of the forebrain, is the most advanced and also the largest part of the brain as well. It has two cerebral hemispheres. We have learned about this from our childhood, very childhoods. We have heard that there is a left side and a right side of your brain. The left side controls the right and the right controls the left, and so on and so forth. A lot of it is hearsay, but yes, majorly these are facts. These are true facts. There are two cerebral hemispheres. Each hemisphere has sub-regions or sub-areas, each of which have their very finely regulated and very specific function, and each of them have to perform perfectly in order for the entire brain to function properly as a whole. So that is how the cerebrum basically looks, and or it is organized. And what we also see is that there is a longitudinal groove which is present between the two, which is like, if you look at the brain, it's not smooth, right? Apart from its surface having foldings, if you look at the top also, it's not a continuous surface. The two hemispheres have a sort of partition, you can say, and that partition is known as the median fissure, which is what has been mentioned here. And it's not that they function independently. While we have heard a lot about how each hemisphere has some role to play, the two are actually connected by the corpus callosum, which is an extremely important part. It's basically like a stack or a band of very thick nerve fibers, and they connect, or they sort of form the bridge or communication window between the two hemispheres, so that they are able to uh, communicate or convey information or transmit messages to each other. And in fact, this is how I was taught about the nervous system as well, where we were told that if in an individual, due to just out of curiosity, if in a person you cut or you break the corpus callosum, the individual would stop functioning. They would just simply turn into a vegetable, because the two hemispheres are no longer talking to each other. So a person cannot survive with half a brain, if that's what you're thinking. So no part or no hemisphere is able to function entirely on its own. That is why the brain is made the way it was, right? So this is an important part to remember. The corpus callosum is the connecting link or the bridge between the two hemispheres. I think I have a picture for this as well. Yeah. So this is the cerebrum. As you can see, we have the left and the right hemispheres. We will talk about the sulci and the gyri as well. But apart from that, these are the different lobes. If you see the brain from the side, we have the frontal lobe, the parietal lobe somewhere in the middle, the occipital lobe somewhere close to the back. Occipital, as you understand, is helping in vision, and the temporal lobe. And what you see at the bottom is the hindbrain, which is made up of those three sub areas or subparts, which, as we are already familiar with, are the pons medulla and the cerebellum. So coming back here, so here we talked about the corpus callosum. We understood that the brain outer portion is majorly, it is made up of cell bodies, very few myelinated axons, and hence it gives rise to the grayish appearance of the brain, as we are familiar with. Often it is said, very loosely said, to be gray matter if a person is more intelligent. So this is the gray matter, what you have commonly heard. And the surface of the cortex, it's a very for lack of a better word, it has a squishy appearance, a sort of very highly folded, very similar, you can think of it to intestines, right? They also have a huge amount or extensive amount of enfoldings which are present there. Similarly, we observe here as well. So these individual folds also have different names, like I said, gyri and sulci. So when the folds are upwards, those are called as gyri. The downward folds are downward folds are called as sulci. Yeah. So individually they are gyrus and sulcus. So after the gray matter, what you see, like at the top is the gray matter. What is at the bottom are the axon tracts and very few cell bodies, which gives a white appearance. So this is, if you think about the brain, the outside is gray, the inside is white. And in complete contrast for the spine, you see the exact opposite. You will see white matter on the outside, gray matter on the inside. Yeah. So my net is a little disturbed. So yeah, apart from that, we saw that how each hemisphere has those four lobes, like we already discussed. And the central sulcus, it separates the frontal lobe from the parietal lobes. And so sulcus, as we have already discussed, they are these enfoldings or these divisions or partitions within the brain. So we have the central sulcus and we have the lateral sulcus as well. Lateral sulcus is for separating the temporal from frontal and parietal lobe, and the occipital, which we said is closer towards the back of the head, is separated by parietal lobes by the parieto-occipital sulcus. So here you can see pretty much the same thing. I'm not going to discuss much about it, but just I'll show you the two sulci as we have already discussed. This is the lateral sulcus, and this is the parieto-occipital sulcus, which is you can see it's not a clear partition, but these are distinguishing the different areas or regions of the brain. So, following that, we were talking about the cerebrum, right? So each cerebral hemisphere has three major functions or three major functional areas. First, it needs to sense. When we say sense, it means it should be receptive to whatever information is coming from outside the brain. So it has to receive information, or here as it is indicated, it has to receive impulse. Where does that impulse come from? It can come from the surroundings, from other parts of the brain, from signaling systems which are operational in vivo, and these are conveyed to the brain via some receptors. Some receptors are able to sense that yes, some information has come in, and it is able to relay it. Secondly, we have the association area. So what does it do? After the information or the message has been received, it interprets what has been received, stores that information, and in response to that information, the brain is able to construct and also deliver a response. Now, this association area, when we say association area, it basically means that if you think of, let me try to switch on my video for just a second. So if you think about it as two mountains, right? You have input and you are generating, the organism is generating a response to that input. So to connect these two mountains or to bridge these two mountains, you need to associate that, yes, this is the information and hence I need to generate this response. So that is the responsibility of the association area. And which is why you can see that this area is involved in memory, learning, and reasoning. So your rational, you understand that when you find an observation, you try to connect it to what you already know. For example, if you think about a researcher, a PhD student, for example, somebody like me, I see some data from whatever experiment I have performed, I get some data, and I have some information based on memory that I have learned previously about whatever I am working on, right? So I need to associate the two. I need to deliver what I have already known and what I'm seeing in front of me. I need to connect the two and I need to deliver a response against it because I'm able to reason. I have the ability to learn. I have the ability to retain the information that I had learned, which is memory, and I'm able to reason it. So this association is taken care of by the cerebrum. And as you can understand, that is an extremely difficult and very important function, and therefore cerebrum is a huge part of the brain, right, as you already know. And thirdly, it is the motor area. When we say motor area, this means that after that information has been understood, processed, processed actually reminds me, if you have gone through the lectures on vesicular trafficking, the process is pretty much the same. So whatever information came in, the antigen that came in, that antigen, not vesicular trafficking, but instead, if you remember about the discussion about MHCs. So when a pathogen enters, that pathogen, the antigens of that pathogen, those are processed, broken down, they are presented, and based on the memory that the immune system of the individual actually has, a response is elicited. So pretty much the same thing. So you sense the input, you associate it, or you reason it out, and in response to that, you execute some sort of action, which is the responsibility of the motor area. So for that action to be executed, there has to be an impulse, and that impulse is carried on to the effector organ, whatever that might be, based on the type of response that you are trying to elicit. So let me turn it off. Yeah. So we talked about the cerebrum. After that, we have the diencephalon. So it is the smaller part. Like we said, the cerebrum is huge. It is the dominating part of the forebrain. What comes next is the diencephalon. It is small and it is present towards the back of the forebrain. So it is covered by the cerebrum. What it is made up of, the diencephalon is not one structure. It basically has these four structures: the thalamus, hypothalamus, epithalamus, and metathalamus. Thalamus is, you might be familiar with this organ or this sub-region. So it is the major part of the diencephalon and it represents the upper wall. What it is doing, it is receiving sensory input or sensory impulses from the body, from all parts of the body except olfaction, which is smell, and it sends or it relays that to the cerebral cortex. So the thalamus is often even considered to be the relay center. And in lower animals, we're seeing that it's the sensory center because the cerebral cortex is not as complicated, or to put it in a better manner, it's not as well developed as you see for adult human beings, right? So basically, the thalamus is relaying whatever information has been presented or has been acquired by the brain. Then comes the hypothalamus, which interestingly enough is a gland, right? What does a gland mean? It basically, it is releasing some sort of material. When we say material, it could be majorly because it secretes or it releases some sort of a hormone or some sort of an information or signal. So it is the master gland, and the pituitary is actually attached to the middle part of the hypothalamus, and towards its anterior surface you would find a webbing sort of structure which is called as an optic chiasm, and towards the posterior part or towards the back of the hypothalamus, in case of mammals we get the corpus albicans. Following that, we have the epithalamus, which is the roof of the diencephalon, and this is where you find the pineal gland. So the pineal gland, if you remember, it's sort of vaguely called as the body clock. It is responsible for remembering or retaining information about patterns that an individual's body under normal circumstances would follow, and when there is a difference from the same, you would see that an immune response is elicited against it. Then comes the metathalamus, which is at the floor of this diencephalon, and it has the medial geniculate body and lateral geniculate body. So each of these, the medial and the lateral geniculate body have individual functions, whereby the medial one, it's responsible for hearing, and the lateral one is majorly related towards the ability to see things or to coming to the next part of the brain, we've covered forebrain. Now we're looking at the midbrain, right? So the mesencephalon, as is more technically called. So it is quite small, and it's the more restrained or more contracted part of the brain. So there are two longitudinal myelinated nerve fibers, which are called the cerebral peduncles or the crura cerebri, which is towards the front part of the midbrain. And you will have some projections or sort of projected regions which are spherical in nature, and they are called as optic lobes at the posterior part of the midbrain. And thirdly, the inferior optic lobes are related to acoustic reflex action. So the midbrain, it's not studied in much depth. It's majorly, it has these structures, and it has more role to play in acoustic reflex action. Now we come to the hindbrain, which, as I've already said, it is extremely important. Why so? So, like you remember, the hindbrain has three regions: the cerebellum, medulla oblongata, and the pons varroa. Cerebellum, do not confuse this with cerebrum. It's quite easy to do so, but they have completely different locations, B functions, and 3 significance. They're both extremely important, but the way they act is poles apart. So what you see here, the first is the cerebellum, second largest part of the brain. What is the largest part? The cerebrum, like we have discussed just now. It is made up of 3 lobes. Lateral lobes, which are large, more sort of spherical in nature, and they're called as the cerebellar hemisphere. So it controls the regulation and the coordination of voluntary muscles. And what it does is it is extremely important in maintaining balance in an individual. When I say balance, it basically means the reason why you are able to stand straight on the ground, you're able to walk straight. All of that is, you need to thank your cerebellum immediately. It has this immense role to play. Second is the medulla oblongata. The medulla is tubular, sort of cylindrical, which is present at the posterior part of the brain. If I come back to this figure, I think you would be able to see. Not very well, but yeah, this is the brain stem, and here you have the medulla oblongata, like towards the absolute back end. You have the pons and the medulla and the cerebellum present there. So it is tubular, sort of cylindrical, which is present at the posterior part. And like we have said, it is extremely important in the involuntary actions of an individual's body, and that is starting some cell secretion, etc. All of this is taken care of by medulla. Thirdly, we have the pons, which is a very tiny, tiny little subpart of the brain, and it's situated somewhere. It's quite deep seated. It's present below the midbrain, and it is sort of it is towards the top part of the medulla oblongata. Sorry. So, it has many nerve fibers. It's very enriched in nerve fibers in transverse as well as longitudinal. And the transverse nerve fibers, they are connected to the cerebellum. And the longitudinal ones, they join cerebellum to cerebrum to the medulla. And it is responsible for the breathing action through the pneumotaxic center. So altogether, they are situated along an axis, which is commonly called as a brainstem. So why I was stressing so intensely upon the role of the brainstem is because if you think about it, A, it's responsible for the body balance. B, like just if you go through the slide very carefully, you will see it is, I wouldn't say controlling, but it is regulating the normal balance of an individual's body. And it is also responsible for breathing. So if you, I don't think anybody might have actually seen it, but if you've seen some sort of enactment of when an individual is, when criminals, they are sentenced to death, now it is done in a different manner by injections, etc. But previously, the approach that was taken was they were hanged, right? They were hanged from some height. And what you would effectively see is that their neck would sort of crack and their head, it hangs loose. What happens there is that this connection, this part or this connection between the brain and the spinal cord and basically the rest of the body, that was broken. What it does is that the connection between the hindbrain, particularly I would say the medulla oblongata and the rest of the body, that is broken. So what is happening is that this extremely important, like the very basic need, you need to respire, you need to breathe, right? So when your breathing is no longer regulated, it's not controlled, when your brain is not indicating to your body that it needs to breathe, what happens is that there is an immediate cutoff of oxygen supply to everywhere in the body, and that individual dies in a few seconds. So while this is quite ruthless, this is effectively what happens there. And in fact, sacrifice, or you can say euthanize animal models like mice or rats, etc. One of the approaches is using some sort of gas, like you basically asphyxiate those mice with chloroform or some other sort of toxic gas, and what happens is they eventually faint, and those mice obviously do not live any longer. But apart from that, another approach that is often used is that cervical dislocation is carried out. The reason why I'm saying in this case, cervical, it's different. It's not, if there's any confusion, it's not cervix, which is found in human females, but instead it's the cervical bone, which if you study the skeletal system, you would find that it's present near the neck, right? The spine basically has different regions, the lumbar, cervical, etc. So this cervical region of the spine, it's basically broken. So like it wouldn't be the most sensitive way to represent it, but if you consider a mice, then if this part is its head, this is the rest of the body, you basically pull the mice tail while you hold on to the head. So the rest of its body gets disconnected from its head, and there is an immediate cutoff of blood supply and effectively oxygen to the rest of the mice body. And as a result, it dies. So they have a different architecture of nervous system in case of mice, but the essence is pretty much the same. So it is controlling, the hindbrain has an immense role in controlling how oxygen supply is regulated throughout the organism's body. So coming back from this rather gloomy discussion, what we will, sorry, my net is very unstable. Uh, what we will do here is that we'll just, you have studied this probably in quite depth, but basically the different regions within the brain, they have different functions, quite obviously, like we have uh, also discussed the corpus callosum, that is why it's regulating the connection between these regions. So here we look at the area where they are located and what is their function. Particularly what you should focus on, I would suggest are two areas. One is the Broca's area and the other is Wernicke's area. So these are often quite confusing because they are, uh, I would say they're similar, but they have different regions, different reasons why they are in place, right? So Broca's area is for speech, and Wernicke's area is for communication, and they are located in completely different regions. Broca's area is in the frontal lobe. Wernicke's area is in the temporal lobe. So you can see that here it is written that Broca's is helping in analyzing speech, whereas Wernicke's is for communication. So you can communicate not just by speech, but by other ways as well, right? You can, like somebody who is unable to speak, people who are unable to speak, they can communicate by sign language or writing or a number of different ways. But that is not what it is all limited to. If somebody's Wernicke's area is damaged, that individual will not be able to, like when I say damage, these damages do not happen very easily. These are very deep-seated areas. Like if you look here, this is obviously a representation of the brain, right? This is the temporal lobe. So somewhere within this lobe is the Wernicke's area, and within the frontal lobe, somewhere deep seated is the Broca's area. So even if an individual is suffering from some sort of traumatic accident, these areas do not easily get damaged. Only under very traumatic injuries, like if somebody has met an accident where their head has suffered the maximum damage or has been the area of impact, only under those circumstances can such damage occur. Apart from that, what happens under a lot of in a lot of situations is when people undergo some sort of surgery, some sort of brain surgery, majorly because they have some tumor within the brain, or they have a glioblastoma or some other sort of tumor, or some sort of removal that needs to be carried out, tissue removal needs to be carried out from their brain. Under those circumstances, when a neurosurgeon, he touches upon these different areas of the brain, only under those circumstances can things like this happen. Which is why under a lot of, if you watch any medical documentaries or even medical shows, you will see that in a lot of cases during these neurosurgeries, usually doctors, they try to keep these individuals open, like awake surgeries are performed where the individual is awake, but brain surgery is being carried out, or that person is being operated upon, their brain is being operated upon. So under those circumstances, those people are asked to do what they do best. If somebody is great at music, they play music, or if somebody sings very well, they're asked to sing or recite, whatever they're talented at, their best abilities, which are the most well retained in their brains, people are asked to do those things. And if the surgery is effectively damaging some part of the brain, which is affecting their abilities, then the neurosurgeon knows that, they need to take a step back and find a different approach. So these two are the major areas that I would ask you to remember well in detail. But yeah, go through the functions of all of the other areas as well. There are not a lot which are mentioned here, so make sure you go through them. Like for example, the occipital area, obviously it is for vision, for sight, and it is at the back of the head, which is why if somebody damages their head, often well, the first thing they lose is their eyesight. They're not able to see for a very long period of time. They're in a comatose state. So that's the first area that gets damaged, which is why this immense stress on wearing helmets, so that you don't damage these extremely sensitive tissues and affect basically the rest of your life. Apart from that, we have the angular gyrus, which is for writing. It's in the parietal lobe. So as you can see, these terms, they are quite similar, like speaking, communication, writing. They're so similar, but these three are located in completely different areas and probably different hemispheres, and therefore the need for communication between the two, stressing on the relevance of corpus callosum once again. But yeah, apart from that, we have the somesthetic area, which is for touching, for feeling, basically the sense of touch. Sorry. The sense of smell is by the gustatory area, and we have discussed Broca's. Apart from that, we have the frontal lobe, the region within the frontal lobe called as the frontal eye field, which is for opening, closing of eyelids. So again, this is not a very, while you can close your eyes voluntarily as per requirement, but you cannot infinitely control or you cannot infinitely not blink your eye. After a period of time, by default, your brain will make sure that you blink your eye, make sure that your eyes are hydrated. And if there is anything foreign which has come into your eye, it will make sure that that gets removed. So these are subtle things that the body has taken over the control for, and it's no longer dependent on your ability to execute the same. So these are the various areas, and the premotor area, if you see here, has the maximum functions. If you look at it from a distance, you will see it is for writing, for movement, for controlling the jaw, tongue, pharynx, larynx, etc. So for speech and communication, again, this area is very, very important. So they're kind of interconnected, but still each has their own role to play. So just go through them. They might be very helpful in answering questions in your, probably in your assignment, but not just that, for your exams as well. So just go through that, and these are important to just know in general. So that covers the brain. The most, or I could say the least understood organ of the body, that is the brain. Yeah, so now we come to the next part, which is the spinal cord. So we had talked till the hindbrain, right? And much in continuation with the hindbrain is the from the medulla oblongata is the spinal cord, and it comes out from the foramen of magnum, and it continues through the vertebral column. So this foramen of magnum, foramen in, again, this brings us to the skeletal system. Oh, sorry. So this brings us to the skeletal system, and when you study the skeletal system, you'll understand that foramen is basically any sort of cavity within the skeletal architecture. So like you understand that vertebrates have a vertebral column along their entire back, right? You have seen the individual stack-like structure that is present within the vertebra, and that is responsible for the curvature of the spine in the way as we know it, which is why we are able to bend, we're able to, move left, right, we are able to execute these n number of different functions that we can. So this continuation is possible because the vertebral column is not solid. It has this cavity throughout the entire length, and it starts from the region where it starts, where it continues from the brain through the neck and towards the spinal cord is the foramen of magnum, which is the main cavity through which all of these nerve fibers are continued and they're carried forward, and from there it connects to the peripheral nervous system as well. So it continues in the neural canal of the vertebral column. And like we had discussed for the brain as well, it is connected by these three layers of connective tissue, which are the dura mater, the arachnoid, and the pia mater. And between these two tissues, which is the dura mater and the vertebra, is the, so how is it coming about? The vertebra or the vertebral column is the bone part of it. The nervous tissue part of it is surrounded by these three layers, and among those, the dura mater is the outermost layer. So the space between those two is the epidural space. So epidural, it's somewhere above Epi, basically the term epi, it kind of correlates to something above, or you can think of it as something beyond the dura. So that is the epidural space. Just for correlation, if you remember epigenetics, I think I discussed this in the session as well. Uh, Something that is above genetics is epigenetics. When we say above, we mean that something which is controlling the gene. So there was a point of time when all scientists thought that the gene is supreme and there is nothing above it. But then, yeah, how each gene within an individual is packaged and controlled regulates how the gene would be expressed. Hence, epigenetics. So similar to that, the epidura is present, or the epidural region is present. So, like I had said that in contrast to the arrangement, I'm sorry, in contrast to the arrangement that you see in the brain, where the gray matter is on the outside, white matter is towards the interior, the spinal cord has the completely opposite structure. So outer part is white matter, inner part is the gray matter. It projects outside, the gray matter sort of projects outside, and it forms the dorsal and ventral horns. I don't think I have a picture for it. Um, I'll try to incorporate it, but yeah, that's the structure of the spinal cord is, it's such that it looks like it is a horn sort of structure, and that is a extension of the gray matter only. So in the root of this dorsal horn, ganglia, which are also cells that are part of the nervous tissue, ganglions, individual and plural ganglia, those are present. So sensory neurons are found in the dorsal root ganglia, whose axons extend and they get embedded into the gray matter. These make synapse with the ventral root neuron. So these are sensory neurons, and so this has been discussed in the lecture as well. I'm not going into it because I think we are already close to an hour of the session, so I'll speed it up a little bit. So motor neurons are found in the ventral root as well. And these sensory and motor neurons, they combine and they come out from the intervertebral foramen. They form the spinal nerve. So we talked about the cranial and the spinal nerves, which are part of the peripheral nervous system. And what the spinal cord is effectively doing is that it is acting as the bridge or the communicator between the brain and body organs, right? It is relaying, or it is carrying forward the reflex actions, and it is providing that path to execute the instructions which are coming from the brain. So that is what you see here. Now, coming to the functions of the spinal cord, as we have already discussed, the major function is the reflex action, right? So reflex, as you understand, is something which is happening by default. You are not particularly thinking about it. It's a response which is coming just by itself. So it's an involuntary action which is executed very rapidly compared to something which you would think about, instruct your brain to do, and then the brain would instruct your organ to do, and so on and so forth. As you understand, there are a lot of mediators which are coming in, a lot of determining factors, and therefore that execution is a lot slower compared to something which is involuntary. So they are majorly of two types. There's a cranial reflex, there's a spinal reflex. So like I said, if an action has to be executed by the brain, so the example given here is excellent. So watering of mouth when you see, or not even see, when you smell something good, right? So many a times when you get some impulse that something good is cooking in your, or something you really like is cooking or is being cooked in your home, you can smell it or you see it, and you can immediately think about how you would. So there are a lot of how should I put it, a lot of stepping stones, or a lot of sub steps which are present in the impulse and the action which is executed, which is the watering of an individual's mouth. That is because your brain is understanding that this is a familiar smell, one that you liked, or this looks like something which you really liked. It is connecting back to the memory, and it is remembering that this is what we felt like the last time we had it. And it is trying to remind you that yes, it felt good, and it gave it, led to the release of a lot of positive, I would say, neurotransmitters. It made you feel good. It released probably dopamine, serotonin, etc. And it made you feel amazing. And therefore, you want to eat it again. So it is going through a lot of sub steps. But When we talk about something which is executed by the spine, which is a spinal reflex, under those circumstances, you cannot afford to take that much time in order to execute a response. And under those circumstances, majorly these are, you can think of them as survival responses, or which have become ingrained as part of an adaptation, as part of an individual's ability to survive. So these actions have to be executed immediately without any thought being given into it. So, for example, what has been said here, when you're getting pinched by a needle, or when you accidentally come very close to a fire, or when you hear a loud sound, or like it's something which sounds like an explosion. For example, if you are suddenly taken aback by some firecracker that might be burst somewhere around you, your instinct is to protect your head, to go down to the ground, protect your head and protect yourself, and to run away from the situation. It's a reflex. You are not thinking about it. So this has become an evolutionary adaptation in order to prolong the life of an individual, and these are survival essential. The others are just the cranial reflexes. Those are taking time because those are not survival essential. Those are actions that are executed in response to a stimulus. So clear difference between the two. And another way to understand it is because when a cranial reflex has to come in, the information needs to go via the spine to the brain, and the response has to come the same route as well, which is why it takes so long. Compare that to the spinal reflex, that information is not taken to the brain immediately. Only after the action has been executed, your brain is then registering that, oh, this are the events that have occurred, and then you register the entire thing as a whole. So that is a spinal reflex. Now we come to the peripheral nervous system. So we talked about the central nervous system in quite a lot of depth. The brain and the spinal cord were the executors of the CNS. But coming to the PNS, which is the peripheral nervous system, what happens here is that all the nerves which started from the brain and also the spinal cord, basically from the CNS, those are all included under the peripheral nervous system. So those include the cranial nerves. When it arises from the brain, when it arises from the spine, it is called as a spinal nerve. So here you can see that reptiles, birds, mammals, they have 12 pairs of cranial nerves from each side. Amphibians and fish, they only have 10 pairs. In case of humans, the first, second, eighth ones are sensory, and these are just sub-classifications which are made: 3, 4, 6, 11, 12. These are the motor nerves, and rest others are mixed. So when we say sensory versus motor, what you basically can think of it as a receptor which will take in the information, which is the sensory one, that, oh, this has happened. And the second set, which are the motor ones, are the ones which are able to associate the information which has come in, that, oh, this is what has happened, and in response to that, this action needs to be executed. So these are two sets of nerves within the peripheral nervous system which have their own predetermined function, and they work in tandem or in concordance with each other after communicating with each other. So we have 31 pairs of spinal nerves. Each is mixed in nature, and it arises from those horns that we talk about from the spinal cord gray matter. So these spinal nerves are also divided into 5 groups, and here you'll be able to see the different regions. The cranial spinal nerve, thoracic. So there's cranial, thoracic, lumbar, sacral, and the coccygeal nerves, because the spine stretches along, like from your neck to the very bottom till your coccyx. So depending on the region where those nerves are present, you'll be able to see that each of these nerves are different. Again, bringing you an example of something you probably might have heard of in real life as well. You might have heard of a lot of people who suffer from pain in their coccyx, right? Who have difficulty in sitting. Often that is because of the pressing of this coccygeal nerve. There is one set of spinal nerves which are present there. So in some people, when the coccyx, it is slightly larger in size, you understand that it does not really have a role to play. It is just an evolutionary component that has been retained. So when in those individuals, their coccyx, it's too extended, it sort of impinges upon the nerves, and as a result, it causes a lot of pain and discomfort in such people. So that is what happens there. So just give me a minute. I'll be back. Yeah. So again, a table that summarizes these functions. Don't worry, I'm not going to go through the details of it, but we'll just understand how they're basically classified. The human cranial nerves, we talked about the spinal nerves just now, and their nomenclature was based on the location or the position along the entire spine. Whereas for the cranial nerves, you can see that it is sort of based on their function majorly. Like if you see here, the olfactory cranial nerves, olfaction, it obviously assists in an individual's ability to smell in olfaction. It is a sensory nerve. Compare that to the optic nerve, also a sensory nerve, but it is helping in vision. It is a part of the system that operates in an individual's ability to see and to interpret what it is seeing. Then comes the oculomotor, which is a part of the midbrain. It is helping in the oculo is something to do with the eye. And when we say oculomotor, it basically means motion of the eye, particularly here the eyeball. Then the trochlear is for eyeball rotation. Then trigeminal basically includes three nerves. From the name, you understand it. It has the ophthalmic, maxillary, and mandibular. So these are controlled by the pons. Again, why it's so important. So it has a lot of different functions. It's important for the chewing motion. So you can often feel it. Like if you chew a lot, you'll be able to see this skeletal movement which is executed. But imagine that the function is um, the signaling that goes behind this function, or the communication that goes behind this function, the ability to chew, it's from right at the back of your head, from your hindbrain. So the brain is a mystery. That's all we can say for now. Then the abducens nerve, the facial nerves, your entire face. It's for all of the expression that you're able to execute. Then comes the auditory nerves. It's in your inner ears. And it's not just for hearing, but you might have studied it in your as part of your course during your school, usually around 10th or 11th standard, when you understand the structures in the inner ear, where you have the inner, the middle ear, the tympanum, and then you study the three bones. From there, you might have studied that the inner ear has an immense role to play in an individual's balance. So that is also a function that is kind of attributed to the cranial nerves, particularly the auditory nerves. Then the glossopharyngeal, yeah, these are cranial nerves which originate from the medulla oblongata. All of them are extremely important. Particularly stressing on this because you should go through this in detail. This, I can say for now that it would be very, very relevant for your exams and for just your knowledge as well. So just treat the medulla as an extremely important organ and try to know and understand as much as about it as you can. So the glossopharyngeal vagus nerve, the accessory spinal is not, it's an important nerve, yeah, but yeah, not that important. But yeah, the glossopharyngeal and the vagus nerve and the hypoglossal nerve are extremely important, particularly the glossopharyngeal and vagus nerve. These are very important for taste, for visceral sensations, for movement. Hypoglossal, usually this term, this hypoglossal glossal terms, they're usually related to movement of the tongue. So glossitis, for example, is inflammation of the tongue. So in case you ever get questions like this, look for terms which are familiar to you, because that's how they are usually, the nomenclature is carried out. So it probably would be helpful for you. So yeah, those were the cranial nerves that we talked about. So we talked about the central nervous system, we talked about the peripheral nervous system, and finally coming to the ANS, which is the autonomic nervous system. What it is, what we initially had discussed, that's pretty much the same here. It's an involuntary, it's the system which controls all of the involuntary or default actions within your body, right? For example, your heart rate. I wouldn't say default. Default is more of a reflex action, which is something which is not entirely in your control, or not that you're not consciously making a decision about. The autonomic nervous system sort of overlaps with it, but it's not the exact same. Because here, for example, your heart rate, you can't control your heart rate, right? It's not a reflex. It needs to continuously keep on beating, but that heart rate keeps on changing based on the type of activity that you are executing. If you are running compared to if you're sleeping. Your heart rate is different, and that is beyond your control. So that is how the ANS works. If you are in conditions where your body is getting extremely warm, a lot of heat is generated, then under those circumstances your body will sweat, it will release that heat, it will release water from the body, and will try to cool the surface in order to reduce that temperature. So that is something which happens by default. Or even peristalsis, for example, like if you consume food, you are not thinking about how that food is going through the entire digestive tract, right? It's a default action, or again using the word default, or it's something you don't have to think about and execute. It's something your body is doing just by itself. So here, of course, if an action is being executed, motor neurons have an important role to play. And these motor neurons are connected to the muscles of your internal organs, particularly the smooth muscles. And it has a very important role in maintaining homeostasis, which is a topic for next week. And we will take it up again in the live interaction session then. And the ANS has two parts. We have discussed this during the classification, which is sympathetic and parasympathetic. So sympathetic is related with intuitive reactions. Let me just read it out. Intuitive reaction, which increases protection of the body in adverse conditions, and that includes the consumption of energy. Parasympathetic system is majorly involved in those reactions where you're trying to conserve energy. So these are somewhat similar yet extremely different. Sympathetic is spending energy just in order to make sure that you are able to survive. Your body is spending energy to help you survive. Parasympathetic is making sure that that energy expenditure is not out of control and that energy is conserved, and it is trying to keep a check on what the sympathetic system is doing. Let's just put it that way. So that is how the ANS basically works. So this was mentioned as a nerve conduction study, which is NCS, a medical diagnostic test, where you estimate the function and the ability of electrical conduction, electrical impulse conduction of the motor nerves and the sensory nerves. So NCV, which is the velocity at which nerve conduction is carried out, that is measured during this test. And for that, electromyography is carried out, where you measure the function of your nerves, of your muscles, etc., and whether there is any spinal nerve compression, other neurological disorders, etc. All of that is studied. So I will, considering whether I should go. Okay, we'll just go through them over thoda on superficially. So for nerve conduction, you have the motory NCS, where you stimulate the peripheral nerve. Electrical stimulation is provided to peripheral nerves, and you record from a muscle. So like, okay, so the nerve is connected to a muscle, right? Because the motor neuron has to execute whatever function is being, is the response to the stimulus that has been provided. So what is measured is that the time, the exact time point in milliseconds at which you are provided with that stimulus, and the time that it takes for that stimulus to elicit the response that is expected. For example, a muscle twitch, you are being provided with a, suppose if you are being tickled, how long does it take for you to jerk away from that tickle? That is the duration which is measured, and that is known as latency. Or if you think about, if you pinch somebody, so the moment that you pinched, from the point that person, or the time that it takes for that person to flinch away from that pinching, that would be the duration of latency. Of course, this is done in a much more regulated and controlled manner, and that is how the nerve conduction test is actually carried out, and the measurement is taken in milliseconds by very sensitive and very well performing instruments. The second was the sensory NCS, much similar to that, and here again the peripheral nerve is also stimulated, but what you are recording is from the sensory portion of the nerve. So what you're measuring is that how well is the nervous system of an individual, I'm just putting it a little vaguely, how well is it able to sense the stimulus that is being sent in. The first one was measuring the ability of the nervous system to respond. Here you are checking, or you are evaluating how well it is able to sense what is being supplied to it. So it is calculated much in a similar way, but here it is measured in sensory amplitudes, which are much smaller in microvolts. The third here mentioned is the F-wave study, which is for which where motor and sensory segments are concerned about nerve conduction velocities in segments of the limb where the F-wave latency decides or derives the conduction velocity of nerves between the limb and the spine. So here the electrical stimulus is applied above the distal portion of the nerve. The nerve impulse can travel in both directions. Either it can move towards the muscle fiber or it can move towards the motor neuron. And based on that, the study, you are basically checking how nerve conduction velocity is being carried. Then came the H reflex study, which is similar, where you are evaluating what is the efficiency with which the stimulus is being relayed or is being conducted between the limb and the spinal cord. The difference here is that impulses that go towards the spinal cord are in sensory nerves, while the impulses that come from the spinal cord are in motor nerves, as we have already studied how nerve conduction is carried out. So it is complex. All of these tests are complex. You need a lot of training. You need very well-equipped testing centers, because these are very sensitive, very expensive instruments. And you need extremely trained practitioners who can interpret what data has been generated. They are often very important. So you don't need to go out of your way and just get a test done. If somebody suffers from a disease, or if they're noticing some symptoms where, their response, their nervous response is anything out of the ordinary, under those circumstances, people usually go for tests like these. And they are non-invasive. They can sometimes be painful because, you're continuously getting stimulated, but they are considered to be quite safe, but they are very important and extremely, I would say, powerful in diagnosing a lot of disorders, particularly where nerve degeneration is a matter of concern. And accordingly, they can help in the treatment of a lot of early onset neurodegenerative diseases, which have pretty much become the norm now. You can find somebody or the other who suffers from some neurodeg disease somewhere around you. So in that way, these are very, very relevant. The EEG is another instrument. I'm not getting into details about this, but yeah, you would, if anybody suffers from some sort of trauma where, it is in any way speculated that their brain function can in any way be affected, under those circumstances, an EEG is performed, which is electroencephalogram, or the report which is obtained is the electroencephalograph, and it is credited by Hans Berger. He connected a galvanometer to a human's head, to their scalp, and you basically have electrodes which are connected. You will find a lot of visuals of this and a lot of television shows and documentaries, etc. So you can see it in the graph here as well, where electrodes are connected to the individual's brain, and what it basically detects is that it is able to detect the change, not change, it is able to detect the brain's electrical response, or the electrical conduction, signal conduction within the brain in response to a stimulus. And it is able to measure it in terms of voltage, and then that data is understood, and you can see how brain conduction is actually being carried out. And in fact, it's an indicator of brain activity, if I just put it very vaguely. And patterns are formed during EEG recording. You have delta, theta, alpha, and beta waves. Delta waves are very low frequency, highest in amplitude. They are the slowest waves. Majorly, you can find them when an individual is sleeping. It is indicative that their brain is working. They're in a peaceful, calm state. Normally, it is also found in babies as well, but it can also be observed when patients are in a comatose state, where their brain is working, but they are not brain dead, but their brain activity is also not normal, if I just put it that way. So delta waves are indicative of that. Theta waves, on the other hand, are higher frequency wave, relatively higher frequency wave. You would usually find them in younger patients, or during meditation, or people who are more stressed. You can find it in them as well. If you see excessive theta waves in individuals who are adults, then it is potentially indicative of some sort of abnormal activity. Third come the alpha waves, which are much higher waves. And awake, but a person at rest has an alpha wave. Um, this was actually the first rhythmic EEG activity observed by Hans Berger. And he was able to see it in the occipital region of the brain. It arises with the closing of eyes and with relaxation. And the more you when you open your eyes and you have more and more stress or more and more mental exhaustion under those circumstances, alpha waves, they decrease. And there are other normal alpha rhythms, which are the mu rhythms, etc., when the hands and arms are just, at rest. Uh, Finally come the beta waves, which are higher, around 15 to 30 hertz. During extreme mental activity, beta waves, they initiate from frontal parietal regions linked to motor behavior, and generally during active movements, they are somewhat rest. Um, Reduced, let me put it that way. A person who is awake, wide awake, and is alert, they have sort of a fluctuating beta wave. The gamma waves are much higher, somewhere around 30 to 100. They are representative of a large population of neurons, and they execute a motor function. And like we said, mu waves are somewhere around 8 to 30. They show that there is continuous firing of the neurons. So what effectively is the function of an EEG is that for a normal individual under different circumstances, so for a normal individual of a certain age group, under certain circumstances, a pattern of brain waves is expected. When in a patient, in an individual who is, say, admitted in a hospital for whatever reasons, when you see that there is a difference in that pattern or there is a deviation from the normal expected pattern, under those circumstances, a doctor or medical practitioner, whoever is there, would try to diagnose what is the issue. Why is that person's brain behaving differently? Particularly, these are performed when somebody has been in an accident, when they have undergone some sort of surgery, particularly brain surgery. Then constant monitoring is carried out to see if some part of the brain might have gotten damaged, or that individual's normal ability might be affected in XYZ way. And apart from that, if a person observes certain symptoms, like for example, here you can see epilepsy, if somebody has a brain tumor, some sort of abscess is there in the brain, if they have difficulty sleeping, insomnia, for example, it's a milder version, but there are more critical brain disorders, where not brain disorders, sleep disorders, where people are not able to sleep. Or if there is some sort of a drug which is affecting an individual's brain activity and basically their nervous system, all of that can be studied using an EEG. So it's an excellent technique, very widely used everywhere in the world, and it has really far-reaching implications in today's healthcare as well. So this was discussed in this week's second lecture, and in the last part, a relatively much smaller part, which we will wrap up soon, and then we can go through the assignment questions before we end today's session. What was discussed in the last part was the nervous system, but basically neurons and their subparts, you can say, or their companion portions which are present within any nervous tissue. So like you can see here, it's the most complex system in humans, and what it does is it basically coordinates all of the different physiological functions. Like we have said, it arises or originates from the ectoderm, receives stimuli, transmits the message, responds to it. So this nervous tissue, when we say tissue, it is made up of cells. What are these cells? Mainly these cells are neurons, but that's not all. There are other cells as well. There are neuroglia, glial cells basically, that are also extremely important. So these, while you won't have a nervous system without neurons, right? They are the functional unit. They are the reason why the nervous tissue is understood, or the nervous system is understood the way that it is, and it is able to execute the functions that we know of today. So it is the functional unit. But under that, like you can see in this classification as well, you have the neuron and you have the neuroglia. So the neuron, it has different subregions, which is like a visual or morphological classification of the neuron, where we have the cyton or cell body, and you have like the center part is the cell body, and the extended regions are the processes. When we say processes, they basically mean extension processes of the neurons, neurites that are present, neurites, dendrites, etc. And together these comprise the nervous tissue. So this is what we just discussed. It has a cell body. This is the cell body or the cyton, and these extended regions are the cell processes which are present. So the cyton is basically the broader part or the more chunky part of a neuron, which has one nucleus. You can see here, it's a uninucleate structure in this entire cytoplasm. And you can find all of the organelles except centrioles. So just off the top of your head, if you think about it, why don't they have centrioles? The very simple reason is, if you know about the function, normal function of centrioles within any cell, they have a role to play during cell division, right? So they help in the the separation of one cell into two daughter cells, the parent cell into two daughter cells. That is not required here. So neurons are end differentiated, which means they do not, under normal circumstances, they do not have the ability to proliferate or to divide, basically undergo cell division. As a result of this, when a person suffers from neurodegenerative disorders that we were talking about, it is a debilitating disease. A debilitating disease is something which does not get better. What you can do is you can offer such an individual, such a patient, with palliative care, which means that you're trying to help their symptoms. You are trying to help them live better. You're trying to cure, I wouldn't say cure, but you are trying to, let me give you an example. For example, if somebody suffers from Parkinson's, if you see an individual who suffers from Parkinson's disease, you see that they have constant jittery movements of their hands, which makes it incredibly difficult for them to do the most basic of life functions, like holding a bottle, for example. They're not able to do that. So you can take care of that symptom, but you cannot cure Parkinson's. The reason is that whatever degeneration, whatever neuronal cell death has occurred in their brain has already occurred, and you cannot rectify that. You cannot stimulate those cells to divide again and just cure things. It does not happen. And it can also potentially never happen unless there is some new discovery which might come up in a few years. It cannot happen because these cells cannot divide. And they cannot divide. One of the reasons why they cannot or do not divide is because they lack centrioles. So it's all interconnected. So coming back here, they do not. So the reason why I went through this elaborate description is so that if this is ever a question that comes up, that what organelle is absent in neurons compared to any other normal living cell within the body, you would know that, okay, it's not able to divide. It is some specific organelle which technically only comes into play during cell division. That is the centriole. But it has some other different cell organelles like neurofibrils, Nissls granules, which are in the neuron. What they effectively do, why they are present here, is because they are conducting impulses in the cyton or the cell body. This Nissls granule, it's not a very distinctive structure. It's not that you would find it in other places. It's because it's like it is a coiled structure which is formed by the ER or the endoplasmic reticulum coiling around the ribosome, and it's a structure, you can say, unique to neurons only. So that is what we see here. And after that, apart from the cyton or the cell body, which is what you see. So this is the cell body, right? We said it has a uninucleate structure, which means there's only one nucleus which is present. Apart from that, you see these tiny little granules, these black colored structures which are present. Those are Nissls bodies. And you have neurofibrils as well. And the extensions that you see from the cell body, like these tiny branching of the cell body that you can see, that is or those are the dendrites. So cell processes, which are cell processes, these are dendrons and the axon. There are multiple dendrites, but there's only one axon. So from the cell body, you have these extended, long, elongated structure, which is the cell, which is the axon. And these axons, okay, so they have we'll come to it actually. They have a covering on top of them, which is the myelin sheath. which helps in saltatory conduction, etc. etc. We'll come to it. So the axon is the longest cell process of a neuron. The others are dendrites, much shorter, much more branched. They form multiple synapses with other neurons, but the axon is the much longer, singular, elongated process from one particular neuron. It is covered by something called as the axolemma. The part where the axon is this axon is arising from the cyton is called the axon hillock. So sort of the junction between the two. If you remember the brainstem and the spinal cord, something similar to that. Not exactly the same, but yeah, just the connecting part between the two. And the ending portion, the terminal part of it, is the branched vesicular structure, which is the telodendria. Telo, this word again, it comes in majorly when you're talking about the end of something. So the telomere is towards the end, so-called end of the genome. And similarly here also you can see telodendria towards the end dendrons, or the back side of the dendrons. Some neurons are often covered by a particular substance, which is a sphingomyelin, which is basically a phospholipid. I think we had discussed this during the classes on lipids in week 5 or 6 sometime, when we were discussing these different biomolecules. At that point of time, we had talked about sphingomyelin as well, and we had said that it is present in myelin sheath, basically the covering, and they are altogether called as a myelin sheath or the medulla. It's a thin membrane-like structure. The myelin sheath is covered by a membrane-like structure, which is the neurilemma here. The neurilemma, or also it's called as the Schwann cells. So this you see, this blue structure that you see, which runs throughout, is obviously the axon. It's the process or the extension of the neuron. On top of it, the coating that you see, that is the myelin sheath, which is made up of phospholipids. On top of that, you have a coating of Schwann cells, and Schwann cells are a nucleated structure. The reason this particular lining or covering is so important is because it helps in conducting impulses. So this neuronal length is huge, right? If a synapse has, or if a impulse has to travel through the entire length, it will take ages for one simple signal to even be conveyed, or one single impulse or stimulus to be conveyed. So for that, nature has come up with this brilliance where if you see, the myelin sheath is never continuous. You can see these like little pockets or little patches of coating, which is interspersed by regions where there is no coating. So there is a hopping of the impulse, or the saltatory conduction of an impulse, which carries it so much faster, so that the response can be executed. So that is the myelin sheath, and that is its extremely important role. So coming to the next part, the neuron can be structurally grouped into different subclasses. So we have unipolar neuron, where from the cyton only one process arises, which is majorly seen in embryos. Like we said, it's the first system within an embryo that develops, which is the nervous system. And this single process can get divided into two processes, of which one would act as an axon, while the other would act as a dendrite. These are pseudo-unipolar in nature. Then come bipolar neurons, where two processes arise. One at one end you have the axon, at the other end you have the dendrite. Often in olfaction, in the retina, which is for vision, in the vestibular and cochlear ganglia, you can find them. Thirdly, you see multipolar neurons, where you have multiple processes. They have one axon, but they have multiple dendrons. Motor neurons, which are the executory component of any response, any nervous response. These motor neurons, they have multiple branches, and they are hence called as, or multiple dendrons, and therefore those are called as multipolar. And particularly for Hydra, for the amacrine cells of the retina, they have no cell processes. These are called as apolar neurons, when they do not have any major, major process or major extension. In those cases, so sorry, those are called as the apolar neurons. And did I get disconnected? Sorry, just a second. Yeah. Then we come to functional classification, like we already have done it. There is the subclassification into afferent, efferent, and interneurons. When we say afferent, afferent basically means it is carrying information from the surroundings to your body or to your nervous system. So it is carrying or conducting nerve impulse from receptors, say for example from the environment, from something in your surroundings to the central nervous system. On the other hand, the efferent ones are those which will execute the effect. So those are the motor neurons. So afferent neurons are sensory neurons. They are sensing what is in the environment. They're bringing that information to the CNS. On the other hand, efferent ones are those which carry the information, or I wouldn't say information, they carry the response which has been decided by the central nervous system from the CNS to the effector organ. So this was the stimulus. In response to it, this is the motion that needs to be executed, that is relayed by the effector neurons or the efferent or motor neurons. Third are the association or the interneurons, which lie somewhere between the two. Often they are somewhere confined within the CNS only. Then we come to the physiology of the nerve. I'll go over this a little briefly. So two main properties are that a nerve needs to be excitable, or it should be responsive to a stimulus. And in addition, it should be able to conduct that stimulus or conduct that impulse. Excitability is the ability of the nerve cell, nerve fibers to enter into a state of excitation in response to the stimulus. And when it is able to transmit or carry forward that excitation in a particular direction, it is conducting it, and therefore that ability is called as conductivity. So here first, the resting membrane potential has been explained, where the cell is not, it's the interior and exterior of the cell are not, for lack of a better terminology, they are not equal. There is a polarization. When we say polarization, it basically means the two environments are different. So the cell interior is always negative, and that negative state under normal circumstances has to be maintained, for which there is an immense regulation or checking that is carried out to make sure that the cell interior is always maintained or approximately maintained under rest at the resting membrane potential of approximately minus 65 millivolts. So it is maintained, and for that active energy expenditure or ATP expenditure is carried out, or active transport is carried out for the sodium, using the sodium potassium pumps, and also some passive diffusion is carried out, but majorly active transport has a really big role to play in it. So here you can see that sodium potassium pump, what exactly happens there is, by expenditure of ATP, the removal of 3 sodium ions from the cell interior, from the neuronal interior or axon interior, along with the uptake of 2 potassium ions. So it is -3 and +2. So what is effectively happening? There is a -1 which is maintained, a -1 charge for each exchange is retained within the cell interior. And because there is a passive diffusion of ions, the active movement is opposed. So that is what you saw there. And this one major contributing factor to this is the rate of diffusion. So potassium ions are a lot more permeable than sodium ions, which means that potassium ions are released a lot faster from the cell. So anyway, their uptake is less. And in addition, their release or their exit is much more pronounced. As a result, effectively, the positive charge inside the cell keeps on reducing and the negative potential can be maintained. So this leads to a net loss of potassium ions from the axon and the generation of a negative charge within the membrane. That has been diagrammatically represented as well. So then the generation of an action potential was explained. So how, when first depolarization occurs, it initiates a nerve impulse, or it initiates a spike, which is the action potential, which is where there is a change in the sodium ion channel, which is a voltage gated channel. So this you see is a resting axon where channels are closed. Why are they closed? Because calcium ions remain bound to them. So under normal circumstances, in a resting axon, these channels are closed. When is an action potential generated? When there's a sudden opening of the sodium gates. What does it do? Sodium enters inside by diffusion. It leads to increase in positive ions, drops the negative potential inside the cell. So sodium keeps on coming in, Na plus is increasing, 3 Na plus is taken in, and the negative potential keeps on dropping. A change of approximately minus 10 millivolt in potential difference from resting membrane potential is known as the spike. And what it does is, after an initial slow entry of sodium into the cell, when there is a difference of 10 millivolts approximately, there is a spike and there's a sudden huge influx of sodium ions into the cell, which leads to the generation of an action potential. First, this negative resting potential is carried out when the membrane is completely depolarized, which is where it reaches that zero stage, voltage becomes zero. And then the potential is developed across the membrane. At 30 millivolt difference, it corresponds to the maximum concentration of sodium inside the axon. So this is the exciting stage. Now, after the cell or after the neuron has been excited, a fraction of a second after this opening of sodium gates, there is a depolarization of the membrane, which causes opening of the potassium gates. So while sodium influx is huge, potassium efflux begins. So potassium is taken out of the cell. And because of this, it changes from the voltage within the cell, it changes from 30 millivolts to minus 65. Again, the neuron becomes sensitive to receiving another round of stimulus and carry out the same process of conduction. Now, after this information has been relayed or after this conduction has been carried out, normal resting potential is restored by expelling sodium, taking in potassium, and this time period that it takes for this restoration, during which a neuron is incapable of responding to another stimulus, is basically known as the refractory period, because it cannot receive another impulse. So this is the duration where it cannot be further stimulated. So that is what you see here. This is a resting neuron, this is an excitatory neuron, and this is an inhibited neuron where after minus 70 millivolt, it needs to restore the original potential difference in order to be stimulated one more time. These last two slides, particularly this last slide, it talks about the synapse and synaptic transmission. So the synapse is basically the junction between two neurons where they are oriented with each other for functional contact. And in the synapse, the telodendria membrane is the presynaptic membrane. The dendron membrane of another neuron is the postsynapse. So if you imagine a synapse, there is one neuron which is sending whatever neurotransmitter it is carrying, and therefore it is presynapse, because from the presynaptic neuron to the postsynaptic neuron, that particular neurotransmitter will be relayed. Particularly, you can consider, say, acetylcholine, for example, would be relayed. And the space between these two neurons is known as the synaptic cleft. So after action potential is dropping, it becomes calcium ion permeable. Calcium enters in the presynaptic membrane, whereby vesicles would release acetylcholine that reaches the postsynaptic membrane, binds with receptors, and develops excitatory postsynaptic potential. So nervous tissue overall has been discussed in quite a lot of depth. We talked about the CNS, the brain and the spinal cord, the different regions of the brain, different regions of the spinal cord, how they are differently executing their own determined functions. How the speed of the response and the detail or the orientation of each of these responses is different. Then came the PNS, which is the peripheral nervous system, whereby we talked about cranial nerves and the spinal nerves. And very briefly, we touched upon the autonomic nervous system as well, the sympathetic and the parasympathetic systems that work in tandem with each other in order to execute the autonomic nervous response. And finally, we understood the functional unit of any nervous system, which is a neuron, and its other component factors as well, and the synapse and how each of these functions can also be studied using EEG and other techniques as well. So that pretty much sums up the discussion for whatever the topics were for this week. So we'll just go over the assignment questions, and after that we can terminate today's class. So this was for the recap. This is an excellent image, so you can go through it for your own understanding. But yeah. So in the assignment, the first question that was asked was about how the human nervous system is working. So it originates from dash layer of the primordial germ layers. So we discussed this in detail, how the nervous tissue is among the first to develop when an organism is undergoing embryonic development. So the nervous system originates from the ectoderm. Uh, from each of these other germ layers also, you can find development of different organs like mesoderm, endoderm, and endomesoderm. Each of these have, uh, each of them lead to development of other organs as well. Then the subarachnoid space, which is filled by the cerebrospinal fluid, is the space between among the different options. If you need better clarity, you can look here how it is. What is this? Yeah, how the subarachnoid space, which is filled by the cerebrospinal fluid, where it is located. If you look here carefully, you'll be able to see that it is located between the arachnoid and the pia mater, right? It's the blue colored portion, which is the question, which is what the question is actually asking. So yeah, the arachnoid and the pia mater, they actually surround the subarachnoid space. So yeah, don't get confused, because this option could be confusing in case you don't actually know the answer, like dura mater or arachnoid. No, it's the dura mater. It's the pia mater and the arachnoid. Between those two, the CSF is present, because the pia mater, if you remember, is the layer of connective tissue which is absolutely snug with the rest of the brain. So between the inner and this metal lining, you'll find the CSF. Then the next question that was asked was about the correct order of the meninges which surround the brain. So if you go from the outermost to the innermost, it's not an option, but if you remember, what you can actually touch and feel is the scalp. Right underneath it is the skeletal architecture of the skull bone, the fused skull. Under that, you have these three connective tissues, which are the dura mater, the arachnoid, and the pia mater, which is absolutely snug with the brain. So I'm pretty sure everybody would get the brain and the skull bone right, but yeah, these regions can often be little bit confusing, but yeah, you just eventually learn. You can find easy mnemonics for this. I'm not mentioning it here, but yeah, the name dura can be remembered as something which is far from the brain, in case there is any confusion. But otherwise, I think diagrammatic representations are make it easier to remember things like this. Then the next question asked about dash layer of the meninges, which is made up of collagen fibers. So if you remember, we talked about how this collagen is one of the toughest materials within the human body, and it is, it helps in making something which is very durable, or it composes things which are very durable, very strong, and which provide a lot of protection. So of course that has to be a part of the skull, and part of the entire head region, which is very protective, and therefore that would be the dura mater, because right underneath the skull you can find the dura mater, as it is a layer of connective tissue. Then sorry, did I miss a question? No. So in question 5, we were asked how the human brain is divided. This is extremely easy. You do not need to get confused, because if you think about it, the human brain has like an insane number of sub-regions. But if you just look at the brain, if somebody handed you a human brain on your hand, what you would see is that it has two parts, right, which are separated by the median fissure, and which are connected by the corpus callosum. So it has two cerebral hemispheres, or two parts. Then in question 6, we were asked that the olfactory lobes are present in, if you remember this, it's a direct question. It was there in the lecture notes. So it's part of the hindbrain. Then we have in question 7, dash is also referred to as the master gland in the human body, just the hypothalamus. I think we'll go through some of these regions in the next week's discussion on homeostasis, how they release certain hormones, certain molecules which are very, very crucial. So the hypothalamus has a has a lot of very important functions to take care of. Then in question 8, we were asked about the main function of myelin sheaths in a neuronal cell is to, this is quite direct, but I'll still go through the options, which is about A, generating signals, receiving signals from neighboring neurons, providing insulation, preventing leakage of ions, or allowing leakage of ions. So obviously option D is not true, but basically myelin sheath provides insulation and it prevents unnecessary ion leakage. So it is, you can think of it sort of as a seal. It's not a continuous seal, but a layer, or maybe even analogous to a waxy cuticle on the surface of a leaf, which prevents unnecessary transpiration, right? Similar to that, you don't want unnecessary ion leakage, and also it's providing some sort of insulation. In question 9, we were asked that the neurons which help in nerve conduction from the central nervous system to the effector organs, so it is relaying information from the CNS to an effector organ, some part of the body, which will execute some motor function. So that part is taken care of by the efferent neurons. The easiest way to remember it is when it has to execute, when it has to execute an effect, that job is taken care of by an efferent neuron. Compare that to an afferent neuron. What gets affected is the afferent neuron, where information is brought to the CNS by them. So these are easy. Whatever helps you study, whatever helps you remember, study that way. Derive mnemonics which help you remember, and it's something you can do for the rest of your life. Yeah. In question 10, we were asked that the resting membrane potential is maintained by the active transport of ions with the help of A, hydrogen ion pumps, magnesium chloride ion pumps, but yeah, we know these are by sodium potassium pumps. So if you remember in the start of the interaction session, I had also mentioned about how during old age, you often see a lot of older people, elderly people, having difficulty in speech, in remembering things, in their overall behavior, which gets affected by a sodium potassium imbalance. So you understand, right, how immensely important the levels and the control of levels of these ions is very, very important. You cannot have an excess of them as well. You can't keep on feeding somebody a lot of salts in order to fill their body with a lot of ions, because that has adverse effects on other organs, particularly on the kidney, which is filtering out, which is carrying out the entire process of excretion. That is immensely harmful. Excess of anything is bad, and particularly excess of such ions, such salts, is very, very detrimental for the kidney and for the excretory system as a whole. So there is a fine balance which is maintained within the body, which is why the stress on healthy diet, despite how difficult it might be, but yeah, you need normal levels of sodium potassium to be present in the body for your brain to function properly. Body is not getting nutrients, it's getting deprived of these basic minerals. Your brain is not able to think. So again, my internet connection is unstable. Anyway, we are almost done with the class. So yeah, those are sodium potassium pumps which get affected. In the final few questions, we were asked which type of neurons are found in cochlear ganglia. I think the example was already given there, but yeah, bipolar neurons are found. We talked about how structurally neurons can be quite different. So bipolar neurons are present in the cochlea. The sympathetic nervous system belongs to the ANS, or the autonomic nervous system. And finally, in question 13, we were asked that the optic chiasm is found on the anterior surface of. This is a trick question. If you don't go through it through the diagram and through the details of the text quite intensely, you would miss out on it. But yeah, in the hypothalamus at the anterior surface, you can find the optic chiasm. So finally, in the last two questions, we were asked that the longitudinal myelinated nerve fibers called as cerebral peduncles are located on the midbrain. It's a trick question as well, but yeah, they're located on the midbrain. And Finally, in question 15, we were asked that which spinal nerve has the same number of nerve pairs such as that of the lumbar spinal nerve. Again, this was another question that if you didn't go through the text in detail, in depth, and it's a memory-based question, I know, but yeah, you need to remember some subtle details. So the answer to this would be the sacral nerve. It has the same number of nerve pairs as that of the lumbar spinal nerve. So yeah, so these were the assignment questions and these were the topics that were discussed for this week. Um, with this, I think I'll end the live interaction session. We are kind of done with the time as well. So I hope you've been caught up with whatever syllabus has been covered till date. So we only have 2 more weeks left, and after that we'll be appearing for your exams, and we'll probably have another live interaction session with the professor and a doubt clearing session. So you can compile all your doubts and we can take them up. You can post questions if those are required, and go through the notes in a lot of detail, go through all of the assignment questions. You might find a few common questions or something from those questions might be quite helpful for you. Perform well in the assignments because those marks are also, they get included in your final grading. So yeah, that's pretty much it. All the best. And with this, I'll close the session for today. And yeah, good night.